Hi, I'm David from the Maths Learning Centre at the University of Adelaide, and this seminar is called Thinking About Data. It's about how statisticians think about data and how they make graphs and summaries in order to help them think about data. Before I get into those graphs and summaries, I want to tell you a few things about statistics generally so that the stuff I say later makes a bit more sense. First, stats helps to answer questions. That's what it's for. And the questions it answers begin by being about concepts. So your research question that you're going to use statistics to help you answer is usually about a single concept, such as how much potassium is there in a banana, or about the relationship between concepts, such as does the variety of banana make a difference to the potassium that it contains. Most research questions are about the relationship between concepts, and most of them are not about the relationship between just two concepts, but are about the relationship between multiple concepts. Now, statistical questions are not actually about concepts, they're about variables. And concepts become variables when you measure them. So for example, with the potassium in the banana, you need to choose how you're going to measure that. Is it going to be the raw amount of potassium, or is it going to be the amount of potassium per gram? And any number of other things have quite complex decisions about how you're going to measure them, such as measuring depression in people, for example. That has a lot of decisions about how you're going to measure it. Are you going to measure it as yes or no, or mild, severe, none, or give them a survey where they have a lot of questions that they score out of 1 to 5 and they get a total score? That's really quite a complex decision. But once you've made that decision, your concepts become variables. So I like to draw concepts as little thought bubbles, whereas I like to draw variables as nice, well-defined rectangles. And so now this question becomes a question that statistics can cope with because it's about variables. A very important thing about variables in statistics, which helps make sense of a lot of what happens, is that variables come in two main types. They can either be numerical, or they can be categorical. Numerical variables are also known as quantitative or interval or scale, depending on who's teaching you your statistics, and they represent numbers. So if you write about an individual or an object under study, if you write down a number, then it's a numerical variable. And sometimes you write down numbers and they're not really numerical variables, and you can tell if a number really is a number by seeing if how far apart the numbers are makes any sense. And I'll get to that in a second. Categorical variables, also known as qualitative variables, are all about categories. So the purpose of them is to put objects or people into categories, and so they tend to be words like male or female, or group 1, group 2, group 3. Now I can tell that the 1, 2 and 3 in those categories are not actually really numbers, because group 2 is not 1 more than group 1, and group 3 is not 1 more than group 2. Groups 1, 2 and 3 are just different. And so you can tell that they're not really numbers because how far apart they are has no meaning. Now, in a minute, I'm going to draw pictures to help me understand how variables are working. And they're going to look a bit like this. So I might put a box with the name of the variable in it, and then I'll put a little picture underneath to say whether it's numerical or categorical. So if it's numerical, I'll draw a little arrow like that with marks on it to indicate that it's like a scale on which we're measuring things. If it's categorical, I'm going to draw a couple of boxes like that to say that we're putting people in boxes. They're the categories that we're putting people or objects in. The next thing is that subjects are sources of variables. So in statistics, the word subject means whatever it is that you're recording information about. It could be people, could be objects. And to a statistician, a subject is nothing more than a source of variables. People are walking around and there's just variables coming out of them, and we record the variables, and that helps us get our statistics. So here's some pictures here. I've got a banana and I've recorded its age, its weight, what variety of banana it is, and the amount of potassium in it. Okay, so that banana is a source of 4 variables at the moment. Or I have people. Perhaps I've got a question about whether chili raises your body temperature, and I've recorded for this person their temperature with chili and their temperature without chili. And I've got a petri dish here. I've recorded what the culture medium is, what treatment I gave it, and I've counted the number of bacteria colonies on day 1, which is numerical, a number of bacteria colonies on day 2, and the number of bacteria colonies on day 3. Now, another important point about variables is that variables can be used to calculate new variables, and the variables that you calculate from them still count as variables in their own right. So for my person, I can calculate the temperature difference between before and after chili, and I can, for the petri dish, I can, from the 3 numbers of colonies on those 3 different days, calculate some sort of average growth rate. And even though they're calculated from the other variables, they still count as variables in their own right. Right. So this next piece of information is the thing that statisticians always ask me to tell. So every time I tell statisticians that I'm going to do a seminar and I say, what should I tell people? They ask me to say this. And what they ask me to say is, information about subjects goes into rows. So when you record data, you normally put it in some sort of Excel spreadsheet, and this is how a statistician wants you to do this. So each person or object will have a row of their own, and all the information about that person will belong in a row. So you see person number 1 here has a gender and age, whether they had chili and a temperature, and I've recorded that in the first row. And person number 2, and they've got all that information too, and I've recorded that in the second row. And person number 3, they've got all that information too, and I've recorded that in the third row. And person number 4 would go in the fourth row, and so on. Now, the reason why statisticians are so interested in you doing this is because arranging data this way means that your computer will be able to cope with it. Computer programs are written in such a way that if the data's not in the computer in the right way, they won't do what you expect them to do. So you need to record data like this and have no other information in the Excel spreadsheet where the data is. Don't record means or any other summaries. Just keep the raw data and everyone will be happy, and the statisticians say that any other mistakes that you might make they can deal with if you've done this. Now that I've said it, I can move on and tell you other important things, but it is actually quite important to the stuff I'm going to be saying later. So let's get into these graphs now. The first kind of graph is the scatterplot, and I think the scatterplot is the most fundamental graph. It's very usual at school or in any statistics course for them to teach you things like histograms and boxplots before scatterplots, because they think that research about a single variable is somehow simpler. But it seems to me that most statistics is actually about the relationship between variables, and so the most fundamental graph is the scatterplot, and I'll tell you a little more about why I think it's more fundamental later. So on the scatterplot, each subject, and that means each row in your data, is a point on the scatterplot. Information is represented by what the point lines up with on the axes, and possibly by colour or shape or other attributes of the point itself. And your aim is to see the variation, so how the variables change, and see past the variation to the overall structure, the overall pattern of how the variables change in response to each other. So here's an example. This data is about a crash test situation. We have cars, and the cars are going at certain speeds, and then we've recorded their stopping distance, how long it takes them to stop when they're going at that speed. And so you'll see the data has, for each car, a speed and a distance, and I'm interested in the relationship between speed and stopping distance. So if they're going at a higher speed, does that mean they have a longer stopping distance or a shorter stopping distance, or what? And so here's the graph here. So this row here is a car which has speed 4 and stopping distance 2, and that would correspond to this point here because it lines up with 4 on the speed axis and 2 on the stopping distance axis. And this row here, which is a car with speed 10 and stopping distance 18, would correspond to this point here because it lines up with 10 on the speed axis and 18 on the stopping distance axis. Now there's some terminology for what goes on these axes. This one is called an outcome variable, also known as a dependent variable or a response variable, depending on who's teaching you. And this one here is called an explanatory variable, also known as an independent variable, depending on who's teaching you. But independent has a specific meaning in probability, which is related to statistics, so I'd prefer to use explanatory. Now it's worth noting that your outcome goes on the y-axis, and normally you try and do it in terms of causation. So speed causes the stopping distance to change, and so we put it round this way. But from a statistician's point of view, we can't actually show causation using statistics. We can only show if things are numerically related. So for them it doesn't really matter, and it's all about what you're trying to calculate. Because if you imagine a crime scene investigation team which comes to a crash site and sees the skid marks on the road and therefore knows the stopping distance, they may want to be calculating how fast the car was going before they had the accident. And so they may be more interested in this relationship. How do you, from the stopping distance, calculate the speed? And now, because the speed is the outcome of my calculation, it's the outcome variable. And the stopping distance is being used to calculate it, so the stopping distance is being used to explain the changes in speed. So it's all about the calculation. Now I'm going to show you some more types of scatterplots in a second, but I want to move on to the line graph first, just to point out how it's different from the scatterplot. The line graph was the first kind of graph to be invented, and you may think that makes it more fundamental than the scatterplot, but I'll tell you why it's not in just a second. Line graphs in general show the changes of things over time, and for that reason subjects are represented on the graph multiple times, because they're there on the graph for each time they're there so that you can see the change in time. Information is still represented by what the point lines up with on the axes, and points for the same subject are connected by lines so that you can tell that they belong to the same subject. But this is quite different to how a scatterplot works. So this graph here is representing the growth of 5 orange trees over time. Their circumference has been recorded on various days in their life, and I've drawn a dot to represent what their circumference is on various days, and I've connected the same tree up with lines. But you'll notice that each tree has 4 dots on this graph, and that's different to how the scatterplot works. The scatterplot only had 1 dot per row. And that's actually a problem because a statistics computer program can actually only deal with 1 row per dot. Let's have a look at how that works. We've got repeated measurements on the same subject. So here's my tree, and I've recorded its circumference on day 118, its circumference on day 664, its circumference on day 1231, and its circumference on day 1582. And tree number 1 would have all of its data in row number 1, and tree number 2 would have all of its data in row number 2. But the problem is that each of those numbers I want to be represented on the graph as a separate dot. In an ordinary scatterplot, which is the only thing a computer really knows how to draw, that first row could only be represented by 1 dot, and so I wouldn't be able to represent all 4 days separately. I need to arrange my data differently so that the computer can draw the graph that I want. So therefore what I need is each row to actually represent a tree-day combination, because that's what the dots represent on my graph. So about a combination of tree on a certain day, I can write down which tree it is, what day it is, and the circumference. So the data looks like this. That first row of data in the original dataset becomes 4 separate rows of data in the new dataset, and about each row I write down what tree it is, what day it is, and the circumference on that day. And now I can draw a graph which has days on the x-axis and circumference on the y-axis, because I have data that has those 2 variables measured in its columns, which we didn't have before. So this graph is actually representing this data. My subjects are tree-day combinations, and I have each tree represented on 4 different days. And what I've been able to do is draw the relationship between day and circumference, also the relationship between tree and circumference simultaneously. And the color has told me which tree it is, and the dot tells me what the circumference is on a certain day. Alright, back to scatterplots. Well, in this situation, it's still a scatterplot, and you might not have seen this before, but this is a scatterplot with a categorical explanatory variable. This research is about guinea pigs. Guinea pigs are given doses of vitamin C. They either have a low dose or a high dose. They're also given different supplements, either vitamin C tablets or orange juice. And we record after a certain amount of time how long their teeth are, because we know that vitamin C is related to gum health. And guinea pigs are one of the only other animals other than humans that don't make their own vitamin C. So here's my data. I've recorded the length of the teeth, what supplement they had, and the dose they had. And I'm interested at the moment in the dose, whether it's high or low, how that's related to tooth length. So dose is a categorical variable. It's words, either high or low. And I can still draw a scatterplot. So this row of the data here, that's a guinea pig which has a low dose and a tooth length of 4.2, which is this dot here. It lines up with low on the x-axis, and it lines up with 4.2 on the y-axis. But let's have a look at this. Look at these 2 guinea pigs. Guinea pigs number 7 and 8. They both have low, and they both have 11.2. So that's about here roughly. But because those two guinea pigs have the same dose and the same tooth length, those two dots are drawn on top of each other, and I can't tell on this graph that they are on top of each other. So I'm going to have to do something a bit more creative to be able to see exactly how much data there is in each zone. What I'm going to do is create a jittered scatter plot. I'm going to just add a little bit of random width so that the points are spread out a little bit, so it's a little easier to tell how many of them there are in each zone. And now those two guinea pigs that were on top of each other are now separated. The reason why this is better is because we can still see all the individual guinea pigs on this graph. That's why that scatter plots are fundamental, because we can see all the individual bits of data on the actual graph itself. So this is a jittered scatter plot, and I can see where the data is a lot more clearly than my ordinary scatter plot. And just to show you how I could get a third variable on this graph, suppose I wanted to know how supplement was affecting the outcome as well. Well, I could add colour to say which supplement it was. And so now I can see simultaneously the effect of dose and supplement. The sort of orangey ones are orange juice and the green ones are vitamin C tablets. And you can actually see, we can see past the variation to see that all of the orange juice ones are higher. We could already see that the high doses were higher, but the orange juice makes it even higher again. Let's look at another scatter plot. What if we had a categorical explanatory variable and a categorical outcome? Now, this research is about people. There's students. We've recorded their gender, their eye colour and their hair colour. Eye colour has been recorded as brown and blue hazel green, and hair colour has been recorded as blonde, dark and red. This person here has dark hair and brown eyes, and so they would line up with dark on the x-axis and brown on the y-axis, and that would be them there. But of course this person here also has dark hair and brown eyes, and so they would be here as well. And so we've got severe overplotting, it's called, where the points are plotted over the top of each other. So what if we add some jitter to this? I'm going to add some random distance to the left and right and some random distance to the up and down, and we'll get this jittered scatter plot for my categorical variables. And now we can see that it's very unlikely for blonde-haired people to have brown eyes. We can also see that there's less red-haired people than others, and we can see that for dark-haired people it seems to be more or less equal as to whether they have brown eyes or blue-green hazel eyes. And let's see what happens. We could add the gender into this and colour by gender, and we can see that, look, the genders seem to be evenly scattered across all of those, so gender doesn't seem to make a difference to hair colour or eye colour or the relationship between them. So one more scatter plot. What if you've only got one variable? We've got one variable which is just the time taken for a geyser eruption. This data is for the Old Faithful geyser in Yellowstone National Park, and as the geyser erupts we record how long the eruption takes, and then next time we record how long that eruption takes, and so on. And so we could draw a scatter plot with no y-axis and just draw all the dots that line up with the x-axis. So for example, this eruption here is about here, and we can do that for all of the dots. But of course it's a little difficult to see where all the data is. There is a big gap somewhere around 3 seconds there in the middle, but it's hard to see how much data there is in the other zone, so we can add some up-down jitter, and that's a little better for seeing where the data is. We can see there's quite a strong clump here and another fairly concentrated clump here, but it's really not as good as the other scatter plots. But all the individual data points are in fact on this graph, which is part of what I was saying about how the scatter plot's more fundamental than the other graphs. But it's not as useful as we would like. So there are two graphs that we can draw to get us a better feeling for how much data there is here and where the data is and how spread out the data is. The first one is a histogram. The histogram shows how much data is in each zone. Now we actually have that in our original one-dimensional scatter plot, which has dots to indicate how much there is, but just so that there's a better way of having a feel for that, what we'll do is in each zone, you've got to pick your zones, so I'm going to pick half-second zones, and I'm going to count how many dots there are in that zone, and draw a box to indicate how many there are, like this. And so in that first zone, under 2 seconds, is represented by this box here, and it goes up to a height of about 50. That's saying there's 50 eruptions in that zone. And the tallest one's here. So it's most likely for an eruption to be between 4 and 4.5 seconds. Of course, your traditional plot doesn't have the dots on it, and so now we have this histogram, which is trying to show where the data is, and we can see that there's a big clump between 4.5 and 4 seconds and a big clump between 2.5 and 2 seconds. There's also the box plot, which has the opposite purpose as a histogram. The histogram is designed to show where the data is, and in each zone you count how much data is there. The box plot, you pick a particular amount of data and you see how wide the zone that contains that data is. So here we have our original data, and we're going to draw this, and then this zone here contains a quarter of the data, and this zone here contains a quarter of the data, and this zone here contains a quarter of the data, and this zone here contains a quarter of the data. And so that's a box plot. There are in fact quite creative ways of figuring out where those end markers are on both ends. Some people put them at the 90% mark, some people do the width of that box and 1.5 of those from the center and only go out to the last data point before that. But your traditional box plot, which was invented in 1977, they go out to the maximum and the minimum. And actually on that note, these lines here have names of their own. The left-hand end is the minimum, then there's the first quartile, because it's after the first quarter of the data, then there's the median, which is in the center of the data, and then there's the third quartile after 3 quarters, and then there's the maximum. Those are what those markers are called, and there's another number, which is the interquartile range, which is how far it is between the two quartiles, so the width of that box. Right, so there's something really important to say about these two graphs, which is the individual subjects are not on those graphs. On the scatter plot, each dot represented one of the subjects. On these graphs, there are no subjects here at all. In fact, the numbers that are recorded on these graphs do not refer to any one of the subjects at all. The median, for example, isn't one of the subjects necessarily, and even if it was, we would only know if it was the median because the rest of the data is there. All 6 of those numbers on the box plot, the minimum, the first quartile, the median, the third quartile, and the maximum, and the interquartile range, are measurements of the whole dataset and not of the individual subjects at all. And back in the histogram, the heights of those bars does not belong to any one of the subjects. All of those heights are information about the whole group of subjects, which brings us to our next point. Variables can be recorded about a group. We've had variables on each subject, but we can take the information about the whole group and write down a variable that belongs to the whole group. So now my subject, in inverted commas, is the group itself. So for example, if I had an experiment measuring the temperature of people, I could write down the median temperature, I could write down the mean of the temperature, I could write down the percentage of temperatures above 37 degrees Celsius, I could write down the IQR of the temperatures, I could write down the standard deviation of the temperatures, I could write down the maximum temperature, and any number of different things I could write down, but none of those belong to any one of the subjects at all. All of those measurements belong to the entire group. Before I move on, I should say that the median and the mean are called measures of center. They're designed to tell you where the middle is. So in some way trying to say where is the whole group. So if I move the whole group as a clump, where's the middle of that? So that's what the median and the mean are designed to do. And the IQR and the standard deviation is a measure of spread. So if I look at the whole group as a clump, how spread out is it? Well, the IQR and the standard deviation do that. So if I'm going to draw a graph of my data, I'm going to have to create a brand new data set where each row represents an entire group. I can't just tack it onto my other data set because the computer's not going to be able to tell whether the new information belongs to subjects or groups. So I need to make a new data set which belongs to the groups. So in this one I've got a situation where I've got a group which had chili and I measured their temperature, and a separate group which didn't have chili and I've measured their temperatures. And so I've been able to clump all the people in group 1 that had chili together, and I've recorded the mean temperature and the percentage of temperatures above 37 degrees. And then in the other group, which had no for chili, I've recorded their mean temperature and the percentage above 37 degrees. This is called aggregate data, and also these are called summary statistics. So let's draw some graphs of group statistics. This is my original graph of my guinea pigs, which was trying to simultaneously show the effect of dose and supplement on tooth length. And what if I aggregated that data so that the 4 different groups, the ones for low and high, the ones for 2 different supplements, have aggregate data? So here we go. Now we have a group of 10 guinea pigs, and about that group of guinea pigs I can write down the dose and the supplement and the mean tooth length. And so there's my new data, and now I have a dot for each group. This is still a scatter plot, but it doesn't have the individual guinea pigs on it. It has the groups of guinea pigs on it. Now we may want to put some more information on this. We have lost a lot of information by removing all that lovely jittered scatterplot that we had before. So what if we wanted to include the standard deviation? So we've got a new data set which has standard deviation, and I'm going to have error bars. So I'll go up by the standard deviation and down by the standard deviation. And that's what that looks like. And it gives us an indication of how spread out each of the groups were, but it's a little bit difficult to read, so I'm just going to shift them over a bit so that they're not overlapping and it's a little easier to see what's going on. So we can see that on average, a group of guinea pigs who all had orange juice will have higher average tooth length than a group that has vitamin C, and also for low versus high dosage as well. So what if it was more a categorical situation? So we've gone back to our eye colour, hair colour thing. So I could create some aggregate statistics and do the percentage that are in the blue, green, hazel category for all my students. So I'm going to split them into groups based on hair colour, and then each group of hair colour is going to have the percentage of blue. And I could draw a scatter plot for that too. And there it is there. Now this is really unusual to draw a scatter plot for this, because remember back on the histogram, we had a whole lot of dots and we wanted to summarize how many dots were in a particular zone. Well, we could do the same here. We had a whole lot of dots here and we want to summarize how many dots are in the blue, hazel, green zone in each of these three groups. Well, normally to summarize how many dots, how much data is in a particular zone, you normally draw a bar. And so this is a bar chart. It goes to the same heights as those dots we had before, and it's a representation of how much data was in the blue, green, hazel spot for each of those hair colours. Now we can also, if we want to, calculate the percentage in the brown category and add that to our graph. And this is called a stacked bar chart because I've just stacked the brown one on top. So they all add up to 100%. It is possible to do this with the raw numbers, but it would be a little bit more difficult to compare because there were so many fewer people in the red group, so I've done it with percentages. So this is a stacked bar chart, and we can still see that it's much more likely if you're blonde to have blue, green or hazel eyes. So they're my graphs of group statistics. The traditional ones are dots with error bars or stacked bar chart if you're doing categories. And there's one more graph I want to draw. But to do that, I'm going to have to talk a little bit about this biggest idea in statistics. So, it begins like this. Your subjects are just some of the subjects that you could have had. And your group of subjects is just one of the groups of subjects you could have had. And therefore, any one variable that you record is just one of the variables that you could have had, even if it was calculated from other variables or recorded on the whole group. Anything that you write down, whether word or number, would have been different if I'd had a different group or a different subject. That's why they're called variables, because they're variable. But it's only one of the things that we could have had. The description of all of the values that could have been and how likely they are is called the distribution. And the biggest idea in statistics is that everything has a distribution. Somewhere in the universe, hidden from us, is a list of all the things that could have been and how likely they all are. And that's true for anything we could possibly write down, anything that we could calculate and then write down. There is a distribution that describes all the things it could have been and how likely they are. And that's really important when we get to things like inferential statistics, hypothesis testing and that sort of thing, because that's all based on comparing what we have to what we could have had. So, along with that big idea that everything has a distribution, is that there are some distributions that we know the name of. If we choose our measurement or our calculation carefully, we might even know the name of the distribution. So if we want to draw a graph of a distribution, well, they tend to look like smooth histograms. For example, if I have an individual guinea pig and I measure its tooth length, I will get a particular number. But if I record all the things that could have happened, for all the possible tooth lengths that could have happened, I can record how likely they all are with this graph here. This graph is a probability density function. The height represents how likely it is to be near that number, and the area represents 100% of probability. So all of the possible tooth lengths covers 100% of the possibilities, and the lower it is, the less likely it is to be in that zone. So this is a picture of a normal distribution. I could record a measurement of the mean tooth length of 10 guinea pigs, and that would be this normal distribution. I've got an x-axis, which is all the possible mean tooth lengths from 10 possible guinea pigs, and it's much more likely to be near 20 than it is to be any further away. And this is a normal distribution as well. Any number that I could possibly record has a distribution as well. So if I wrote down the standard deviation of tooth length, it's got this distribution, and that's called a chi-squared distribution. Now, only some of the things that we write down have a distribution that we know the name of and have a formula that tells us how high that graph is. Everything has one, we just don't necessarily have a formula for what it looks like. And so we choose measurements like the mean and the standard deviation because they have a much more well-defined formula for how that curve looks. And that's all the graphs that you might want to draw. I mean, there are others, but these are the basic and most fundamental ones. And this is how statisticians think about data.